I'll introduce. Hi, everybody. I'm Paolo Cassano. Welcome to the MGH Brain PBM rounds. And today is February 3rd, 23. It is my great pleasure to introduce Dr. Brian Bicknell, a scientist, trained microbiologist, and also marine biologist, who has bravely left academia to devote himself to study photobiomodulation and the impact of photobiomodulation on the microbiome. Thank you for being here, Brian, and please take it away.
Probably works better if I unmute myself. Okay, thank you for the invitation to, and I've just, thank you for the invitation to present here. I am not a clinician. I am a scientist. We'll present a couple of case studies at the end, if we have time. I should, first of all, declare that I'm a co-founder, shareholder, and scientific advisor to Symbix, and we're using these Symbix devices on the abdomen to help Parkinson's disease. So whenever I say Symbix, you can just disregard that and think generic device, I guess. Okay, why are we looking at the gut microbiome? We started thinking about this because of the difficulty of getting light into the substantia nigra, where the center of Parkinson's disease is. Even if you're using transcranial, you're not really getting light to the areas that are of interest. So you're really looking at a remote treatment, even with transcranial photobiomodulation. And this was begun at Sydney University with John Mitrofanis and Jonathan Stone, and they did a helmet experiment with their Parkinson's disease model. That helmet experiment, they put aluminium foil over the head and shone the light on the mice. And what they found was that they were getting improvement in Parkinson's symptoms and neural regeneration, even though they weren't eliminating the head. We joined their team after they'd done their mice and monkey experiments. We did some mice experiments. We looked at specifically the microbiome to see if we changed that, as well as changing the symptoms of Parkinson's disease. And it kind of makes sense to target remotely for Parkinson's. There's a well-established gut-brain axis, microbiome gut-brain axis in Parkinson's and other neurodegenerative diseases. And there's a direct connection from the enteric nervous system to the brain by the vagus nerve. And there's also a circulation connection. And we know that microbes produce lots of metabolites, and a lot of those metabolites are recognized by our body. And actually, a lot of our metabolites that we produce are recognized by the microbiome. So the two-way connection between the gut and the brain and the microbiome. And we know that light does affect the microbiome. There's a circadian rhythm that the microbiome has, as well as the rhythm that it has with the feeding cycle. And why is that not? Okay. So what is the microbiome? I guess everybody knows basically what the microbiome is, but microbes are really very, very good at colonizing surfaces and growing on surfaces and making use of any nutrients that are there. So any surfaces that we have that are in contact with the external environment will collect microbes, microbes will grow there, and that microbiome, or that microbiota, will be on all of the surfaces that are in contact, and that includes our oral cavity, your respiratory tract, the urogenital tract, the skin, the gut, anywhere we're in contact with the outside environment. And we think of the microbes that grow there as the microbiota. And in this talk, I'll be mostly talking about the bacteria because the archaea, the fungi, the protists, the viruses, not a lot is known about those, but we do know a little bit about the bacteria now. So what grows there is called the microbiota. What we think of as the microbiome is the interaction between our genetics and the microbial genetics. We acquire our microbiome at birth, and some people think before birth. That develops through our ages. It sort of stabilizes about 3 years and then slowly develops. So all the milestones of development, the microbiome changes. It develops as we get onto solid food. It develops depending on what sort of food we eat as teenagers. And one of our big problems at the moment is the Western diet and the amount of, I'll say, junk food that we eat, and that high sugar, high fat content really affects our microbiome. So the microbiome that we have now is very different to the microbiome that people would have had 100 years ago or in other less Western diet countries. So it's affected by a little bit by our genetics. It's affected by our lifestyle. It's really affected by our diet, because what the microbes are doing is eating what gets down to the gut, the colon. And what we're talking about really is the microbiome in the colon rather than the small intestine or other parts of the gastrointestinal tract. It has a huge impact on our immune system. We're constantly challenging our immune system with our microbiome. Environmental factors also affect the microbiome, so toxins, the type of food we eat, as I said, the pesticides that are on our food that we eat, smoking, air pollution, all affect the microbiome. And antibiotics and other medications also have a great effect on the microbiome, and antibiotics particularly because they're designed to kill microbes. So when they get down to the large intestine, they do what they're meant to do and they start killing bacteria. And so that really affects our microbiome. Okay, our microbiota outnumber our own cells by about 1.3 to 1, depending on how you count the bacteria and the microbes. There are about 9 phyla within the gut microbiome, 300 odd genera, thousands of species, and probably tens of thousands of strains. And each of those strains is doing a different thing. They're growing on different food sources, they're interacting with all the others, and they're producing lots of metabolites. Now, most of those strains are uncultivated. We can't grow them in the lab. We don't really know what they do. The whole study of microbiome is only possible due to next generation sequencing, where we take the genes, we extract the DNA, we take the genes and we sequence them, and we compare them to bacteria that we know, and then we extrapolate what those new bacteria do. And the important thing is that the microbes contain about 150 times the number of genes that we possess. So each of those genes is capable of acting, producing proteins, producing metabolites, and those metabolites interact with also the microbiome, the bacteria and other microbes that are there, and also with us. So they produce a lot of metabolites that directly interact with us. Now, a lot of thought has gone into what's a good microbiome and what's a healthy microbiome, what's a balanced microbiome, as opposed to a dysbiotic or bad microbiome. Now, I have to say there's no such thing as a good versus a bad microbiome. It's really the balance of what's there. So in a very healthy microbiome, you'll probably find bacteria that are bad, inverted commas, bad. What we've done in the past is we've looked at a healthy person, we've looked at their microbiome, and we said, okay, these bacteria are found there. So they're associated with good health. And in the same way, we look at diseases like Alzheimer's, Parkinson's, gastrointestinal diseases, and we say, okay, we find these bacteria here, they're probably bad bacteria. Now, on top of that, the best bacteria for our health produce short-chain fatty acids. So if you look at the list of bacteria I've got there as inverted commas, good bacteria, Facilibacterium, Bacteroides, Bifidobacterium, Rosiburia are all short-chain fatty acid producing bacteria. And short-chain fatty acids are very important because they feed the cells that line the gut. And if you have a nice, good food source for those cells, they're happy, they have good tight junctions, you don't get leaking of our gastrointestinal tract into our tissues. Akkermansia is an interesting one. That doesn't produce short-chain fatty acids. What it does, it degrades mucin or mucus. What that does is stimulate more mucus to be produced, and so that's recognized as a very healthy bacterium. It's being looked at as a probiotic. At the moment, most of our probiotics are Bifidobacterium and Lactobacillus, but Akkermansia is being looked at as a potentially good probiotic. Clostridium, some Clostridium are good, and if you look on the other side, some Clostridium are bad, potential pathogens, and that's because Clostridium were named when we were looking down microscopes and they all look like Clostridium, but we found that they actually live in different parts of the phylogenetic tree. So the some types, the 14A and the 4 type Clostridium, produce short-chain fatty acids. The stricto sensu Clostridium on the other side, like Clostridium difficile, are potential pathogens. What we have on the other side, the bad, inverted commas, bad potential bad bacteria are potential pathogens, actual pathogens, and lipopolysaccharide producers. So they're mostly proteobacteria like E. coli, Klebsiella, Salmonella, and you probably know that LPS or lipopolysaccharide is the outer cell wall, or the outer membrane of these bacteria, and that's very antigenic and very inflammatory in our bodies. So they're potential pathogens. They're usually associated with disease conditions, and the same with the Clostridium, Staphylococcus, and the Streptococcus. So generally found in dysbiotic microbiomes. Okay, the gut microbiome has a profound effect on our health. It's very important for our immune system, as I said. A lot of our immune system is centered in the gut, and it's being continually challenged and probably setting up low-grade inflammation our whole lives. And that's probably not a bad thing. We're improving our resilience. We're looking at, which bacteria are friendly, which ones are not. We're producing antibodies against the bad bacteria. But as we age, our immune system ages, our gut integrity ages, and our microbiome ages, it becomes more of a problem. We change our microbiome, we change our inflammation, and a lot of diseases really are based on this systemic inflammation that increases in pathology and increases with age. And as we age, also our medications tend to increase, and a lot of those, as we'll see, have a lot of influence on the gut microbiome. Okay, there is a gut-brain axis, there's a gut-respiratory axis, a gut-skin axis, a gut-everything axis, really, because everything that the microbiome is doing is affecting many, many parts of the body. There's many diseases that can be linked with a dysbiosis in the gut. I think I've said most of this already. A lot of immune system being challenged immediately. Dysbiosis leads to a breakdown in the blood-brain barrier, so you get more metabolites, bad metabolites reaching the brain. There is a two-way communication. The brain talks to the gut via the vagus nerve and enteric nervous system, and the gut talks to the brain. And there has been an increasing amount of research into the gut-brain axis and looking at the gut-brain axis as a way to affect brain disorders, and especially neurodegenerative disorders and things that don't have other avenues, or very many other avenues for treatment. And that includes metabolic diseases, type 2 diabetes, cardiovascular diseases, metabolic disorders, many, many diseases. So there's a lot of research going into how to affect the microbiome and change the gut-brain axis in a good way. Okay, we produce lots of metabolites. I think 95% of our serotonin is produced in the gut. A lot of dopamine is produced in the gut. Alpha MSH, tryptophan precursors are very important in terms of producing metabolites. Short-chain fatty acids are probably the most important ones, and of those, butyrate is the most important. So a good, healthy microbiome producing butyrate means you're going to have healthy gut lining, which means you're going to have less leaky gut and less metabolites moving into the tissues. So it's associated with multiple diseases, obviously gastrointestinal diseases, IBS, IBD, metabolic syndrome, type 2 diabetes, cardiovascular disease with trimethylamine and oxide, and particular bacteria that are associated with cardiovascular disease, liver disease, chronic liver disease, non-alcoholic liver disease, respiratory diseases. It's linked with COVID. Good microbiome means you're going to have a better outcome with COVID. Sarcopenia, arthritis, and neurodegenerative diseases. And I mean, you would expect this with that very strong gut-brain link. So we think about the microbiome as an avenue to affect these diseases. I think I've said most of this already. Dysbiosis can be caused by many things. One of the main things these days is diet. I'm not sure what the rate of obesity is in the USA, maybe 35%. We're looking for confirmation from Paolo.
Yeah, I think I'm not an expert on obesity, but I would say so, if not even higher.
Yeah, and overweight is much, much higher, and maybe well over 60% of Americans are overweight, and that is linked with dysbiosis. So just being overweight or being obese means you're going to have a dysbiosis in your gastrointestinal tract, which sets you up for all of these other diseases. Alcohol will change the microbiome. Heavy drinking of alcohol or alcohol dependence will change the microbiome. Medication and, as I said, antibiotics are a very big factor in the microbiome, and we'll see that a bit later. And we think of this dysbiosis as being epidemic, and it's pandemic because more and more countries, more and more peoples are going to a Western diet. That high fat, that high sugar, and low fiber is definitely correlated with dysbiosis. It's also endemic, which means it's everywhere and it's not going anywhere soon. It's characterized by reduced diversity, and that's the main characterization of dysbiosis. It's characterized by reduction in short-chain fatty acids, which means a reduced mucus layer, reduced gut integrity, which leads to a leaky gut, increased inflammation, possibly even pathogens crossing across the gut barrier. Definitely lipopolysaccharides from those proteobacteria, those gram-negative bacteria, and increases in trimethylamine and oxide. So it becomes a vicious circle, a positive reinforcement of bad things happening. The more dysbiosis you have, the more leaky gut you have, the more inflammation you have, which leads to increased dysbiosis in the gut. As I said, antibiotics are particularly bad. It can take a number of months to reestablish a healthy microbiome. And often the post-medication microbiome is not the same as the pre-medication microbiome. It can lead to opportunistic pathogens, Clostridium difficile infections. There's an interesting case of rifaximin, which is been shown to be anti-inflammatory. It's good against particular types of bacteria, but it's actually seems to improve the microbiome overall. So that's been called a eubiotic rather than an antibiotic. Other medications, metformin has been shown to improve the microbiome. So short-chain fatty acid bacteria are improved, Akkermansia is improved, and diseases like obesity, type 2 diabetes are reduced. Other anti-diabetic drugs are also good for the microbiome. Possibly statins are good for the microbiome. There's a lot of animal evidence that statins are bad for the microbiome, but there's a little bit of recent evidence showing that they actually might be anti-inflammatory and good. Probably the worst of the medications that I've listed here are the proton pump inhibitors, really widely prescribed, incredibly bad for the microbiome. What you're doing is breaking down the proton pump in microbes, which is how they get their energy. So very bad for the microbiome. They're really over prescribed and really shouldn't be prescribed so much, I think. Laxatives, obviously very important, and selective serotonin reuptake inhibitors and other psychotropic drugs, steroids, non-steroidal anti-inflammatories, even aspirin will adversely affect the microbiome. In Parkinson's disease, very strong gut-brain link. Probably not so much looking for a particular pathogen causing the gut-brain, causing the disease, but it's thought that a lot of Parkinson's can start in the gut, and that's constipation is an early sign, often an early sign of Parkinson's disease. Whether there's a dysbiotic or dysbiosis signature is really hard to say. Different studies will say this is the bacteria or these are the bacteria that are causing or associated with Parkinson's, but there are lots of studies and they all have kind of different signatures. So hard to say that there's a specific signature. Dysbiosis in this case is associated with alpha-synuclein accumulation in the gut, and it's thought that that directly transfers to the brain. So in animal studies, you cut the vagus nerve, then you reduce or eliminate Parkinson's. In some countries where they use selective vagotomy for ulcers a number of years ago, that reduced the rate of Parkinson's disease. So the gut is a potential therapy. Interestingly, if you look up there, Akkermansia and Bifidobacterium are actually increased in Parkinson's disease. We said before that Akkermansia is a good bacterium in most cases, but in Parkinson's, it's increased. In a number of studies, it's been increased in Parkinson's. The same as Bifidobacterium, which is one of our probiotic bacteria. So the question is, why are these bacteria that are normally considered good, why are they increased in Parkinson's disease? We can improve, or we can try to improve the gut microbiome by diet, reducing processed food, reducing non-food foods like manufactured foods that don't have actually any food in them, such as if you look at the Impossible Burger, look at the ingredients, there's no food in that, or no real food. There's ingredients and nutrients, but not food. I don't want to say don't eat them, but actually I'm saying that, don't eat them. Reduce processed foods, reduce sugar, reduce fat, increase vegetables, increase fiber. The Mediterranean diet has been shown to be good. Interestingly, the ketogenic diet has also been shown to be good, which is kind of diametrically opposite. Fermented foods are very good. It's not really effective for Parkinson's. Prebiotics, fermentable fibers, so soluble fibers, and also phytonutrients can help the microbiome. Probiotics, there's less evidence for, good evidence in animal models, much less so in human trials. There's very many trials going on for probiotics, but the results are really quite equivocal. Fecal microbiome transplants. This has been very good for Clostridium difficile infection. It may be effective for IBS. There are trials for Parkinson's, Alzheimer's, MS, obesity, and so on. There is not great evidence yet. There are some case studies that show good results, and there are ongoing trials, as we said. Interestingly, it's just been approved by the FDA for Clostridium difficile, and the TGA, which is the Australian equivalent, has been approved for a number of conditions, including autism. But there are many, many off-label studies where people are using fecal microbiome transplant for patients with these diseases. Lifestyle, you can change your microbiome if you strict enough, reduce your stress, improve your sleep, expose you to microbes, go out and play in the dirt. That's very good for your microbiome. Try not to age. That would be good to improve, keep your microbiome good. Most of these things don't actually change Parkinson's disease. In animal models, we have been using PBM. There are animal models showing that using PBM, mostly in mice, does change the microbiome. This last one down here, the duodenal dual wavelength one, is interesting because they put a fiber optic down into the duodenum and directly irradiate the microbiome or the bacteria that are down there. But all these other ones are using transcutaneous PBM. We did the same thing. We looked at a mouse. It was a Parkinson's disease model. They were healthy mice, so before they were given MPTP, we gave them either red light, LED red laser, 808 laser, or sham, and we did next generation sequencing. These are the microbiome results we found. Down the bottom are the replicates of firstly red, then infrared, multiple doses, the placebos or no laser, and then the single doses. So what we're doing is trying to just look at that and see where are the changes, which is quite difficult. If we look at the beta diversity, we can see that 660 red 3 times a week showed a change in the beta diversity, so a change in the microbiome structure from no light or from pre-light. 3 times a week with infrared also changed the microbiome structure, and a single dose actually changed the microbiome structure as well, significantly. So
Brian, just a quick interruption here. So can you repeat to where, what were the sites of irradiation in these animal models?
Yeah, sorry, the one that we did was on the abdomen, but in a mouse model, if you irradiate the abdomen, you're really irradiating a lot of the mouse.
Right.
And most of those studies are over the abdomen. So they're looking at irradiating the gut.
Right. And so we can assume that the gut was directly irradiated in these animal models.
I think we can assume that. I think certainly infrared will get into the gut, and red probably in a mouse will get into the gut.
Thanks.
Okay, these are the bacteria of the genera that we saw. We found changes, significant changes in just one genus, and that was Alabaculum, sorry. And Alabaculum is down there. So it's a very small proportion of the microbiome, but it was enough to significantly change the balance in the microbiome. Just to put that into context, we've changed or increased the numbers in the microbiome of Alabaculum by 10,000-fold with multiple doses of infrared 808. That's a beneficial bacterium. So we started with healthy mice, but we improved the microbiome in healthy mice with increases in Alabaculum, which is not a human microbiome constituent. It's similar to Akkermansia in mice, so it does the same things. It produces short-chain fatty acid, it renews the mucosa. It's also increased with metformin in a metformin model, and it's found in increased numbers in fiber-rich diets for mice and decreased in obese and fat mice. So we've done that a number of times in different models. We've only published that one study, but we need to do this in people. So we looked at this in our Parkinson's for our first two Parkinson's trials. In Adelaide, we did a trial with a very small number, only 12 participants. We did transcranial PBM with the V-Light, and we did abdominal PBM with our Symbix laser. That's an abdominal protocol and C1, C2 in the neck. It was done 3 times per week for 12 weeks. That's what we found. And again, what we're doing is looking at the difference between before and after for each of our 12 participants. And you can see, if you look at it, you can see that there are changes. You can see that purple one is very big now, whereas before it was quite small. Our red one was bigger and it's getting smaller. So there are changes. If we take the average of those, we can see those changes a bit more clearly. Reduction in that one, increase in that one, increase in that one, increase in that one, and so on. If we do that statistically, we don't get statistical difference. We don't have enough people to get statistical significance. We get a significance in the firmicutes to bacteroidetes ratio, which is, some people think, an indicator of good gut health. So that improved significantly. But the other increases in bacteroides and some other bacteria that produce short-chain fatty acid, non-significant. We also got a reduction in pathogens, again, non-significant. Interestingly, those early bacteria that I talked about, the Faecalibacterium and the Rosiburia, we didn't change those ones. And they're really quite important bacteria for short-chain fatty acids. We didn't change those. We did a second experiment just with abdominal treatment, and we found essentially the same things. That was also published. Sorry, the second one has not been published yet. The second one is on the way to being published. So I'll just do a couple of quick case studies. How am I going for time? Not too bad, I guess. Tell me to stop if you want me to stop. No, keep going.
We want you to keep going.
Okay. I feel like I'm speeding up to try and get through. Okay. First case study was a cancer patient. So she did the PBM treatment after her cancer immunotherapy. So we took triplicate samples this time to try and get a bit more of a feel, because microbiome can change with time, and from not day to day, but it can change with time. It's actually quite stable over long periods, but you can get changes with, as I said, diet and medications. So we took triplicate samples this time. We did baseline, we did after her immunotherapy, and then we did, that was pre-treatment, then we did 12 weeks of treatment, and we did it again after the PBM. What we saw this time was a change in alpha diversity, which means the richness of each of the microbiome. So the numbers have changed significantly between post-treatment and the 2 pre-treatments. And we saw differences between the samples, which is the beta diversity. So we saw big changes with this one person. And these were significant changes, but again, just one person. Again, they're the changes. That's baseline, triplicate, pre-treatment, triplicate, and then you can see there's a big change in the post-treatment. What we saw was an increase in the Akkermansia, a large increase. We couldn't detect the Akkermansia in the first 2 sample sequences. So the baseline and the pre-PBM, no Akkermansia were detected, but they appeared after PBM, and they appeared in large numbers. So it was really indicative of a healthy microbiome. In this case, we saw increases in Bifidobacterium, Faecalibacterium, and Rosiburia. So all of the good bacteria, all the bacteria we know produce short-chain fatty acids and are associated with a healthy microbiome, all increased significantly. And we had decreases in these so-called bad bacteria. Certainly, Eggerichia is a potential pathogen. Para-Eggerichia is a potential pathogen. That's Clostridium is a potential pathogen. Diarrhea is a potential pathogen. Okay, so we saw big changes and we saw significant changes But again, just one person, and we published that one. Our second case study was from our Parkinson's trial, where we did both abdominal and transcranial treatment. Now, if you listened to Anne Leavitt's talk last week, you would have met Margaret and seen her progression and her improvement over 1 year, 2 years, and then 3 years. So she's a good responder to our treatment. She came with severe GI symptoms. She had constipation and diarrhea for 5 years, such that she really thought she couldn't be on the trial because she couldn't travel for more than 15 minutes without finding a bathroom. She had anxiety, loss of confidence. We used the full abdominal, neck, and transcranial treatment. What we found was that her symptoms improved, which Anne talked about last week. And she had all her severe symptoms disappeared within that 12, I'm just talking about the 12 weeks now. And she went into full remission from her GI symptoms after 3 weeks, no more constipation, no more diarrhea. And that continues to this day, which is 4 years. They're the sort of improvements that we saw in her motor tests. So improvement in walk speed, improvement in stride length, improvement in timed up and go. And in this case, the quicker you do it is the better. So a drop is good. Timed up and go cognitive was about the same or a little bit improved from baseline, but the others were improved, and they continue to improve over 1 and 2 years. That's her microbiome for those 3 times, so baseline, 4 weeks, and 12 weeks. Again, really hard to distinguish what's happening. If you take out the top 10 or so bacteria, that's what it looks like. And we're seeing changes in even after 4 weeks, we're seeing a change in the microbiome, getting bigger still. This one's decreasing, this one's improving. What I've done is taken the individual bacteria and put them into 2 graphs of, inverted commas, good bacteria. So what are recognized as good bacteria, and mostly they're improving. This one here, which is doesn't appear to be on the list, but it's it's not a Parabacteroides. I will have to get back to you on that one, but that is a short chain fatty acid producer, and that's decreasing. The others are increasing. Even these ones are increasing a small amount. It looks like a small amount, but it's actually increasing 3-fold or 4-fold. And we're increasing our Rosiburia, Faecilibacterium, and our Akkermansia for this particular patient. You remember that overall we didn't improve these, but for this particular patient, we've improved those good bacteria. That's our Akkermansia, which is increased in Parkinson's, and we're increasing it more. I'm not sure if that's a good thing or a bad thing. There are so-called bad bacteria. Bifidobacterium, which is, again, usually a good bacterium, but is increased in Parkinson's. We're decreasing that with our PBM treatment. Streptococcus, obviously a bad bacterium. Bifidobacterium, Blautia, a bad bacterium, and Doria, usually considered a bad bacterium. Those are decreasing mostly with her microbiome. Our case study 3. I think I said that antibiotics, a history of antibiotics is predictive of Parkinson's risk. So an epidemiological study in Finland showed that there was an increased risk with long-term antibiotic use. This person had his onset of Parkinson's coinciding with a jet lag event plus broad-spectrum antibiotics called prostatitis. And that was continued over a long period. And 3 months after that, he came down with Parkinson's symptoms, including micrography, and had a foot drag, had a mask of Parkinson's, low speech, and so on. So he was diagnosed with Parkinson's stage 3. Oh no, stage 3. He was treated with the PBM on the abdomen and neck only, so no transcranial. He was in the second group. He improved. We could see his improvement dramatically over 3 weeks. His foot drag disappeared. He lost his mask of Parkinson's. He had expression. His voice was better. He then went on antibiotics for a chest infection, and everything went backwards. He self-reported his symptoms were worse. Subjectively, we could see the foot drag again. We could see the expression disappear from his face, and his balance got worse. His MOCA actually didn't decrease. It continued to stay at 30. This is his, say, static balance, which is dramatic. Static balance, they put one foot in front of the other, they close their eyes and see how long they can balance. His static balance with his eyes open when he started was 30. So one foot in front of the other, eyes open, he can balance. After 4 weeks, when we tested everybody, this is 1 week after his antibiotics, his static balance had decreased dramatically with eyes open. That began to improve again after his antibiotics and when we continued the light treatment. We didn't test him at 3 weeks. We tested everybody at 4 weeks. You can see his walk test again slightly decreased at 4 weeks and then began to improve after. His numbers of steps or step length didn't change too much, but then improved to 12 weeks. So there's a big change with his antibiotic use, giving him Parkinson's and then reproducing the symptoms of Parkinson's that we had improved to 3 weeks, but we don't have objective data for that, just anecdotal data. And let's see. That's his microbiome for his good bacteria. That's what happened at 3 weeks before his antibiotics. This was at 3 weeks after his antibiotics, and then over, that should be week 12, sorry. So what are we seeing? We're seeing not much change, antibiotics, and then his good bacteria increase. His Facilibacteria increases, short-chain fatty acid producer increases. His Coprococcus, which is a short-chain fatty acid producer, increases. Not what we would expect. We'd expect those good bacteria to go down with the antibiotics. His bad bacteria, well, some have increased, some have decreased. His bifidobacterium decreased after antibiotics and then started to rise again. I guess what I'm saying is this is not clear cut. Even with this one person who had a dramatic change with his microbiome, dramatic change with his symptoms at 3 to 4 weeks, his microbiome has changed, but not in the way that we would predict if the microbiome was directly affecting those symptoms. Our fourth case study was an Australian singer who he was a singer in Boy and Bear. You probably haven't heard of that band, an indie band. He was so bad with his IBS that he couldn't travel, couldn't record, and so the band went into hiatus, didn't actually disband. He met one of the gurus of FMT, and that improved his symptoms, so he could start recording again in the studio. He then found a match for himself, and he employed a poo roadie who travelled around with him and allowed him to tour. So he was getting fecal microbiome transplants daily to be able to tour. We gave him a prototype device. This was before we developed the full protocol. He was using that, and his symptoms improved to the extent that he could tour, travel, and without too many problems. So it improved his symptoms above and beyond what he's getting with FMT. I was actually interviewed for this Rolling Stone article, but they didn't leave me in. They thought the poo roadie was much better copy. So I'm in there and light was used as well. This is his microbiome pre and post. When I got his microbiome, I didn't know who he was, and I didn't know that he was on FMT. I looked at his microbiome and I thought, this doesn't make sense. I don't understand this microbiome. And when I found out that he was an FMT recipient, then it all made sense. What we see in the microbiome is not what we typically see in either a healthy or a dysbiotic microbiome. You can see it's dominated 0.4 or 40% with Bacteroides. So it's really hard to see what's happening down there. But Bacteroides, as I said before, is a good bacterium. It's a short-chain fatty acid producer, so it's a good thing to have in your microbiome. If I take away that one, then we can see what's happening down in that lower end. These, again, are the good bacteria, or potentially good bacteria, and mostly they're kind of increasing with our 12 weeks of treatment. The Anaerostipes, which is a good bacterium, short-chain fatty acid, is decreasing dramatically. But all of our other good bacteria are quite good. This one is interesting. This is Morella. It's not usually found in the gut microbiome. It's usually an oral microbiome bacterium. There's not a lot known about it. It is associated with a number of diseases like obesity and chronic liver disease, but it's also associated with improvement in IBD. So we can't really say it's a good or a bad bacteria, but in our case, it's being reduced by the PBM. These are our potentially bad bacteria. This one is called very catchingly S24-7. It's a family of bacteria rather than a genus. Not much is known about it. It is associated with obesity and metabolic syndrome, so we think of it as potentially a bad bacterium. That's our Morella again. And these are the other bad bacteria we've identified. The Clostridium sensostricti, which is the bad, potentially pathogenic bacterium, which was present in his gut at 0.2% and hence decreased. And then we have Megamonas, which is slightly increased, and that's associated with obesity, type 2 diabetes, and so on. So again, it's not a clear message. I have to say that microbiome is really at its infancy. We're really only scratching the surface with microbiome. But we think that we have a way of affecting the microbiome. We can certainly do that in animals. Looks like we're doing it in Parkinson's. It looks like we're affecting it in a good way. It's one of the few treatments that can really potentially affect the microbiome with these diseases, with these very intractable degenerative diseases. So we think it's a good potential mechanism. We certainly are pursuing it. Is it a primary or secondary result of PBM? Are we improving Parkinson's and then that improves the microbiome? Or is the microbiome directly involved in the improvement of our symptoms of Parkinson's? We need to find that out. The mechanism, why does this work? In our group, we have 7-8 people in our group. Everyone will have a different idea of what the mechanism is. So we need to chase this down. My personal idea is that what we're doing is reducing inflammation. That inflammation is having a positive effect on the gut lining. That improves the microbiome. The improved microbiome will have a positive effect on the gut lining, and you have a flow-on positive reinforcement or positive effect. That is all I have to say. I think I went too long, so I cleverly didn't leave enough time for questions.
Thank you so much, Brian. I have a quick question, that really interesting topic and beautiful discussion overall. So I might have missed this, but were there any non-abdominal treatment, so none on the abdomen in humans, with proof of change in microbiome?
We haven't done that. I would love to do that. I would love to try to see what the systemic effect is, and if we could do it on the tibia, the sternum, other places, or even transcranial, and see what changes in the microbiome. That would be a really interesting thing to do. We haven't had the opportunity to do that. I mean, I suspect that there would be changes in the microbiome. I don't know.
Okay.
But certainly, I mean, one of the things we get with our IBS people and our Parkinson's people is remission from their gastrointestinal symptoms. We get very good remission from constipation and diarrhea. So I think the gut protocol is actually quite important, even if just for that.
Got it. I see already a question from Attila.
Yes, thank you very much for this. Did you observe or measure any change in the GI motility and any change in the absorption patterns? Or I don't know how you could measure that with. I wonder.
No, we haven't. We are connecting with a gastroenterologist now, and certainly that'll be on our agenda for next time. That's very important. And we haven't measured any other markers of inflammation or these were very preliminary, and we were looking at Parkinson's essentially, and then the microbiome as well. So yeah, we have a lot to do.
Few more minutes. I see a question from Jeff.
Yeah, hi, this is Jeff from Boston. A couple of questions. When you did not see effects, do you think it was related to the dosage of PBM that was actually received, or it was like, well, this just is not effective? Okay, that's one question. Mm-hmm. The other question is, do you think there's differential effects of PBM on different bacteria, so that some go up, some go down? Yeah. And then my third question is, given that they're all together, do some bacteria influence other ones, like upregulate, downregulate, so that your picture gets a little muddy because of the different direction that they're going?
Yeah, in the last question first, there's a huge interaction between all of the microbes down there. And so anything you're doing to one bacteria would affect other bacteria. In terms of penetration, we don't get the light to the gut. Going through the abdomen, going through the skin, the fatty tissue, muscles, no way we get our light into the gut. So we don't have an direct effect on the bacteria. So it has to be a downstream effect. And I'm sorry, I forgot your second question.
Do you think there's a differential effect of light on different bacteria?
I think so, but I think that's controlled by the body. I think the body tells, because the body is happier, then it encourages the good bacteria to grow. So if you take the bacteria out, put them in the lab and shine the light on them, yes, there is actually a differential effect. Some bacteria will increase and some bacteria will decrease with infrared light. In fact, E. coli, which we think of as one of our bad bacteria, that increases with infrared light. It encourages the growth. And that's not surprising because, mitochondria are really bacteria. So if you affect the mitochondria, you can certainly affect bacteria. But again, I don't think we're directly affecting the bacteria. We're affecting the body. The body's telling the bacteria what to do, or, encouraging the good bacteria. And once you start getting good bacteria, then that shifts the balance in the microbiome because all the bacteria are talking to each other.
And then maybe what I have you on a fourth question would be, do you think that if you monitored these changes for a longer period of time, you would see some different effects? Because sometimes with the clinical data, we see that, some of the effects seem to have a delayed onset in terms of their manifestation.
I definitely agree with that. We have taken samples from our people who were going on for 2, 3, and 4 years. They haven't been analysed yet, because there's people who are doing the treatment for 3 years, 4 years. They're still doing it 3 times a week, every week, because they're staying better, so they're continuing to do it. So it would be really, really nice to monitor these people over a long period of time. Not many studies have actually done that longitudinal monitoring.
Yeah, that'd be great.
But again, we're talking about really low numbers. I need enough money to do a really big study. I need thousands.
Yes. Those are hard to do, those large studies, and it's hard to do them well.
Absolutely.
I think we got to to the hour. Thank you all. Thank you so much, Brian. Very thought-provoking, very interesting topic. Thank you all for being here today.
Thank you, Brian. Thanks, Paula.
Thank you.
Bye.
Thank you.